Okay, now I'm going to show you a specific way to create a phylogenetic tree from original data. And in this particular case, we're going to be doing a parsimony method that looks at what are called reduced matrices of shared derived characters. So let me go through each one of these steps. We have 5 different species here. One we're calling the outgroup, and that's going to be used to determine what are the ancestral states at each of these 7 nucleotide sites. So we have nucleotides at 7 different base pairs. So 1, 2, 3, 4, 5, 6, and 7. And here we're trying to determine what's the phylogenetic relationships based on parsimony and these shared derived characters of A, B, C, and D. Okay. So the first thing we do is determine a matrix. This actually should be a matrix of shared derived characters. And basically this is a pairwise assessment of the number of shared derived characters in common between each one of these species. So how did we get these numbers? So we get these numbers by going up to each of these tables. So let's deal with A and B first. Okay. So what we have here is what we said that A and B share one derived character state in common. And we can go back up to the table and see which one it is. So here's nucleotide site 1. Both A and B have a derived character state C. So that's 1. You can see here they both have G. Okay, all 3 have G, so there's nothing there. And then here we have T as the ancestral state, and A has the ancestral state, but B has the C, which is the derived, but it's not shared in common between the two. Nothing has changed here. Nothing different between these, nothing different between these, and again, B has the derived character state. So A and B, in this particular case, share a derived character state in common only at nucleotide site 1. So it's just put a 1 in there. A and C is similar. Okay, A and C have that shared derived character state in common. Nothing different here. They don't share anything there, nothing there. This is obviously an ancestral state that has the derived state, and nothing is shared between these two. So these two only have one derived character state in common. Okay, and you can figure out what it is for A and D. So basically what you do, you set up this matrix and you determine this upper part and how many characters are changed in common. So let's look at B and C as an example, and then we'll go to C and D. How did we get 3? So B and C share 3 character states in common, derived character states. So here you can see there's a character state shared in common and it's derived, so that's 1. B and C, here's another one where that's a change from T to C, so that's another one, that's 2. Nothing's changed there, nothing shared in common there, nothing shared in common there. And they both have C's, which is a derived character state from the ancestral, so that's the 3rd nucleotide site that they have the shared derived state in common. Okay, B and D have the same. Okay, now let's go to what do we find for C and D. What C and D says is those 2 species have 5 derived characters shared in common with one another. And we can see that here. There's 1, there's another one, that's 2, there's 3, it's derived. That's a 4th derived character changed from A to T, and C changed from G to C, so that's the 5th derived character state in common. Okay, so now we have this original matrix of the shared derived characters. What we do is we pull off the one that has the highest, and hopefully you'll have ones where they're the highest. Sometimes you might have A and B having 5 too, so you would pull that off at the same time. So what you do is you're saying that C and D are actually the most derived. They have 5 shared character states in common, and C and D is what you pull off first. You then develop what's called a reduced matrix, and this actually is the first reduced matrix. And here what you do is you now use C and D, but as a composite, and you basically use the average of these. So A and B, you still have A and B in here, but now you kind of combine C and D together. So A and B is up from here. You still have 1. Okay. The C, D, and A is basically the average of these 2. C and A, how many do C and A have in common? They have 1. D and A have, up in the original, have 1 in common. So you take 2 divided by 2. So they share 1 character state in common. Okay. Kind of the average. Okay. Now, let's look at how did we get three character states shared in common, derived character states shared in common between B and kind of C/D. So here what we do, we'd go to B and C, take the B and C. What do we find up here? B and C is 3. We take B and D, and what do we have? B and D is 3, so a total of 6 divided by 2. They basically share three shared derived character states in common between B and the composite of C and D. Okay, so in this matrix, the pairs that have the highest number of shared derived character states in common are C and D and B, and what we do is we then pull off B to get that in the tree. And obviously the only one left is A, so all we have to really do is then pull that off, and this is the phylogenetic tree that we get using this parsimony, using shared derived characters. We can then map these character state changes onto the tree, kind of like we did previously. Okay, so let's kind of do the first one. Nucleotide site 1, the outgroup is G, and all 4 species have C, so we put the change at character state 1 to C, and you could do G to C, however you want to do it, occurred at the base of this tree, and all of them have that. Okay? Now we can look at 2, right? This is an uninformative character. They all have G, so that you don't map anything onto that tree. Let's look at 3. The outgroup has T, the A has T, and B, C, and D have C. So here are B, C, and D. So what we say is in the common ancestor along this branch leading to B, C, and D, it changed from a T to a C at character state 3. And obviously you can do it for these other characters, 5, 6, and 7. Obviously 4 isn't changing. And you should be able to get these results. And what you should find from this tree is, C and D share these 2 derived character states in common. The common ancestor B, C, and D share these 2 shared derived character states in common, and they all share this in common. And so basically what we have are basically 5 steps. What we will do in class on Thursday is I'm going to give you a different tree than this and ask you to map the character state changes onto a tree using this matrix, and we'll do that as a little extra credit. Okay, so be ready to do that, and we'll do that as a group problem. Okay, now let's kind of finish up on phylogenies, and what I've been showing so far are fairly simple things because we only have 4 taxa, right? We only had 4 species. And what you can see is the number of species or taxa that you have in a phylogenetic tree, or it could be the number of populations that you have. So when you only have 3 taxa in a tree, there's only actually the possible number of what are called unrooted trees is only 1. If you have 4, you can only get 3. But as you increase more and more species, you get this exponential increase in the possibilities of the potential number of unrooted trees that you can find. So obviously, if you're dealing with 2 million possible trees out of 10 taxa, you're obviously not going to do this by hand. And there are lots of very complicated, very sophisticated computer algorithms for determining phylogenetic trees using not only parsimony, but what's called maximum likelihood, to all sorts of other methods. But one of the things that we often want to do, we want to develop phylogenetic trees, and we want to know how much support there is for particular branches in trees, especially as we develop hypotheses. So I'm not going to go into all the details of this, and this is what's called a bootstrap analysis, in that, and basically what you get is, again, you have 5 species, you have different characters. The 1 or 0 just represents species 1 at character A has some value of 1, 2 has 0, and these are just binary character states. So you basically construct a character matrix, which is a list of characters and character states. And basically what you do in bootstrapping is you actually just kind of eliminate particular characters and often duplicate other characters, and it's called resampling. So you kind of pick a new set of characters at random with replacement. And what you can see is actually what happened here is in this sort of random process with replacement, these resampled characters are not, you don't get all of them again, but you actually get 3 C's are included in this matrix, 2 F's are included, and some of these characters are left out. Anyway, so I don't want to belabor this too much, but you actually create a new matrix from these characters only, and what you can see here is you have these 3 C's, they're all the same, and there's a C down here, and here's an F, and they're all the same in terms of what you find in each species. And this is basically what this creates what's called a replicate of the characters. And you repeat this many times to create what are called additional replicates. And in most computer programs right now, you can often create 10,000 to 100,000 bootstraps. And what I'm getting to is I'll show you how we use that to help us actually test hypotheses when we use bootstrap, because it tells you something how well supported particular branches are or clades are within a phylogenetic tree. Okay, I'm not going to belabor this, ignore that. Anyway, the key thing that I want to point here is when you do all this bootstrap, you do 10,000 replicates of alternative trees and you do all these simulations. Basic thing what we're trying to do is what we're saying here is that out of those 10,000 replications, species A1 and A2 and A3 always came out as a monophyletic group 90% of the time. So if you did 10,000 bootstrap replications 9,000 times, A1, A2, and A3 always came out together, being grouped together. And the take-home message is anytime you have values of this particular branch and these three species always being together is 90, that's a pretty high probability that these groupings are real and they're strongly supported by the data. Okay, and so we use this number to say, it helps us to understand that, in fact, A1, A2, and A3 are a monophyletic group. They're well supported by the data, and they represent a real group. So how do we use this in real life? Okay, so here's a real simple hypothesis, the Theria hypothesis versus the Marsupionta hypothesis. So there was some debate, and this has all been resolved by DNA sequence data. And what I'm going to show you in the next slide is based on that. But a lot of people suggested that perhaps the relationships among these three taxa, placentals, marsupials, and monotremes, you could have different sort of sister taxa relationships, and the squamate reptiles were basically used as an outgroup. So the question is, under the Theria hypothesis, the idea is that monotremes, the platypus is an example, are ancestral to the placentals and marsupials, and the placentals and marsupials share a common ancestor. And obviously marsupials and monotremes are primarily found in Australia, but it suggests that, in fact, placentals and marsupials are sister taxa, and monotremes are the ancestral. The other hypothesis, the Marsupionta hypothesis, since both of these are found in Australia, the argument was that marsupials and monotremes share a common ancestor, are sister taxa, and placentals, including placental mammals, as an example, elephants are outside. And so what it suggests is the marsupials and monotremes would be, if these were sister taxa, would support the Marsupionta hypothesis. If placentals and marsupials are well supported as a monophyletic group or a well supported clade, then this would support the Theria hypothesis. And the take-home message is from DNA sequence data, and we're not going to go into all the details, but the basic idea here is that the Theria hypothesis was extremely well supported. So here, the various bootstrap supports for these various clades, and obviously these are all the placentals. They're well supported, the eutherian placental mammals, 100% of the time. They all group together as a monophyletic group, as high a support as you can get. Okay, the key point here is the Theria hypothesis would be where the marsupials and the placental mammals, here's the blue, here's the kind of pinkish, that clade, that ancestor right there, 100% of the time, marsupials and placentals in the bootstrap analysis were found together. So this is 100% bootstrap support for the Theria hypothesis, and the monotremes fall out. So the basic take-home message from all this DNA sequence data is the Theria hypothesis is well supported. Placentals and mammals 100% of the time are grouped together. The Marsupionta hypothesis is rejected. Okay, so that's kind of how we use bootstrap to help us test various kinds of hypotheses and how we can use this to sort of discriminate between hypotheses using phylogenetic trees. And this is being used a lot in current evolutionary biology. Okay, so as I mentioned, be ready to come to class to map the character state changes on an alternative tree from the previous example. And I will see you on Thursday.